So thank you for inviting me today. I'm going to talk about the genetics of intracranial aneurysms. And so first I would like to disclose that I have nothing to disclose. And IA is a very complex phenotype caused by genes, environmental factors, and their interactions. We know some of these risk environmental factors such as smoking, a very important one. And then we do know now some susceptibility loci. I'm going to talk about their identification today. But these interactions will be critical, so that will be kind of the bulk of the common complex forms of intracranial aneurysms. And then it's worth noting that about 5% of all of these cases are associated with inherited connective tissue disorders. So it's very important to ask about the family history of these connective tissue disorders. And so this is just giving you an overview about the similarities and differences in the different types of aneurysms. And now if we look at I'm going to focus on the IA there, you can see that the prevalence is about 2%. And then if we think about the genetics there, so we can see that there is clearly an increased risk for relatives of siblings of these patients. So we are talking about relative risk between 2.5 and 7. And also if we look at the prevalence of among first-degree relatives of these patients, it's 10. So these are high risks. And so what are the risk factors for rupture of IA? So this was one study published a couple of years ago in stroke, and that was a large study, and they identified that these are the important risk factors. These are kind of the key things that, of course, when we talk about IA, that we are interested, what are the risk factors for rupture? And that's also something that we would like to think about in terms of the genetics, so that this is the kind of the main focus. And now how can we identify genes for complex traits such as intracranial aneurysms? So typically what we do nowadays is that we do a genome-wide association analysis. So in that analysis, we will screen several thousands of variants throughout the genome and test those variants for association with the trait. And then we can get significant peaks given the multiple testing. So we have to take into account the multiple testing there. And there, after that genome-wide analysis screen, so then we do a replication study. So the key peaks, the key markers there are then we try to replicate those in an independent study sample. And after that, we can do regional analysis, extensive sequencing of the region, and look for the linkage disequilibrium structure among the variants, and then finally do some functional studies. And so what are the typical study samples for complex traits, genetic studies of complex traits such as IA? So we have these are the most typical ones used. So we have cases and controls, or we can select families. So we can perform family-based association studies. And there we typically collect at least the parents and the proband. But then in the family-based association studies, there often is the difficulty to get the both parents, and also it would be perhaps more a genetic approach to start with families where there are multiple affected individuals. So you can see that it easily leads to kind of much more complicated collection process than collecting just cases and controls. And then, when we think about the case control studies, that is now the main form to do genome-wide association studies. it may be difficult to establish that when the association arises, that what is the direct causality between the factors that we are investigating. And it's critical, totally crucial to think about the distribution of age, sex, and ethnicity between the cases and controls, because those issues can cause stratification bias. So that's very important to focus on this. And in the family-based association, they are kind of a safe method in the sense that they have been developed exactly to circumvent this problem of related or relevant controls, especially if we use mixed populations, such as a study sample from the Los Angeles area. And so there in this setting, so the internal control is then derived from those alleles that are not transmitted to the affected individuals. And so I said that we are using this genome-wide association. So the reasoning why these were studied for complex traits was that there were papers showing that these association tests are much more powerful statistically than the typical linkage analysis when we try to detect alleles of modest effects. And also we will perform these using single nucleotide polymorphisms because the genotyping platforms for these type of markers are much easier and more cost efficient than the ones for the microsatellite markers. So these are different types of variants that we have throughout the genome. And so this is also an unbiased way regarding what is the function of the variant and where is it located. So we can kind of detect all different types of variants. And also, as I said, so this has been made possible by recent genomic advances. So the technology is now allowing us to genotype in one array up to even more than 1 million markers at the same time. And so this is one of the examples of a meta-analysis of genome-wide association studies for lipids. So we have different traits here. We have HDL cholesterol, LDL cholesterol, and triglycerides. And then this genome is screened throughout the chromosomes. And we will have some interesting peaks here. So they are implicated, and they show here in this particular study, they show us which ones are the previous lipid genes that peak, but they also have some peaks that are totally new peaks for lipids and that have never been implicated for lipid traits. So that's the beauty of the genome-wide association studies, that you can also identify totally novel genes that were not connected to these traits previously. And this is a key region that was identified for coronary heart disease. Here we have the risk interval. It's about 58 kilobases long, and we have several associated variants here indicated by these arrows, and then they are in tight linkage disequilibrium, so they kind of are redundant markers. And then if we look at the structure there, we have a couple of genes there, and then we have, so here is again the risk interval, so but nothing really like here, so the signal is between the genes. And this region has been a very strong signal for coronary heart disease. The mechanisms are not fully clear as of yet. And interestingly, the same region is also associated for IA.